thank you the organizers for having me here today. And I have to confess, I didn't know who Louise Harel was before I came here today, but knowing that she was such a remarkable woman, I'm really honored to be here. Well, thanks to Antonin and Igor that did a very good introduction of non-encoding RNAs, I won't need to really go through that part, but what really is clear to all of us now is that our cells are replete with RNA molecules that have the potential to modulate or interact with different cellular pathways, and in an active or a passive way affect gene expression at different levels. As you can see here, there are non-encoding RNAs among them that have been characterized and known to be functional in all these levels. And in my laboratory, in particular, is interested in understanding how these non-encoding RNAs can affect the biology of cancer cells. And to do that, we have paid a lot of attention to a very important cancer pathway, the p53 pathway. As you know, p53 is the most important human tumor suppressor. It's a transcription factor that can integrate different signals that come from damage that occurs to a cancer cell, and it activates a tumor suppressor program that leads to the arrest of that damaged cells, of the death of the cell in case this is, the damage is too extensive and the cell has to be eliminated. So what we found is that non-encoding RNAs can be transcriptional targets of p53. p53 activates directly the expression of a set of non-encoding RNAs that serve as effectors of some of the aspects of the p53 tumor suppressor response. We have done that, and also other labs have been working on this, and now there is a body of work showing that LINC-RNAs play a role in this pathway. I can show you, for instance, one of these RNAs that we have been studying for the last years. This is a LINC-PINT, it's a p53 regulated RNA. As you can see, p53 binds to several loci in this gene, and it's activated by DNA damage in p53 wild-type cells. What we found is that this LINC-RNA acts as a tumor suppressor, and it has the ability to inhibit cellular invasion in liver when we are expressing it in colorectal cancer cells. And what was quite interesting is that when we analyzed this LINC-RNA, and this work was done in collaboration with Igor, we found that it has a highly conserved region that is conserved in vertebrates, and this region is sufficient to do to induce this phenotype of inhibition of invasion of the cells. And inside this conserved sequence, we found a small element that when mutated, it impairs this phenotype. And also, very interestingly, we found that this element is responsible for the binding of this protein called PRC2 to this non-encoding RNA, which seems to be an important element of the function of this RNA in the regulation of genes that inhibit tumor invasion. But beyond this LINC-RNA PINT, we found that p53 is able, as I say, to activate directly a set of LINC-RNAs that we have summarized here in the table. And we have found that these LINC-RNAs that are p53 targets in colorectal cancer cells serve as a tumor suppressor signature that is able to distinguish the cancer cells from the non-cancer cells, indicating that this is really a feature of cancer. But as you know, p53 is mutated in most tumors, and for this reason, we also were interested in knowing what happens in cells when p53 is mutated and they are subject of DNA damage. Under these conditions, the cells are highly proliferative, and they also have increased genomic instability. And they induce the expression of genes that are related to this phenotype. These are the type of protein-coding genes that are induced under DNA damage conditions in p53 knockout cells. But also, this DNA damage leads to the expression of some London-coding RNAs. And this is one of them that we characterized a few years ago, called CONCR. We name it CONCR, but it's the antisense of this protein-coding gene, DDX11. And we found that in p53 knockout cells, this LINC-RNA is induced after DNA damage. And this is happening in a cell cycle-dependent manner. It's regulated in the cell cycle, and also transcriptionally regulated by C-MYC. Consistently with this, we see that it's overexpressed in different types of cancer, and in breast tumors, for instance, the expression correlates with a bad prognosis of the patient. But what was really interesting to us is observing that when this RNA is knocked down, is eliminated in the cells lose the cohesion between the sister chromatids. This is a quite striking phenotype that we observe, and that correlates with the levels of this London-coding RNA, as you can see here, when we use different strategies to downregulate its expression. So how is this cohesion established in the cells? Well, what is known is that the establishment of the cohesion between the sister chromatids is tightly linked to the replication of the DNA. And there are several factors that are involved in this process, like one of these, the protein DDX11. That, if you remember, happens to be the protein that is a neighbor gene of CONCR. This protein, DDX11, is known to it's a helicase that helps to solve stall replication forces sits on the lagging strand of the DNA, and when there is some stress in the replication and stall fork, this helicase comes in place and helps to solve it. So we found that this effect that we find when we eliminate the London-coding RNA CONCR from the cells is rescued by this protein. So if we overexpress the protein, but not the catalytic mutant of this protein, we can recover the cohesion of the sister chromatids. And the first hypothesis that we had when we observed this is that this London-coding RNA CONCR would be, in fact, regulating the protein DDX11. And to analyze this, we did a lot of experiments looking at the RNA levels of DDX11, the protein levels, as you can see here, when we knock down CONCR, don't change. And also we look at the chromatin in the environment, it's not affected. So we couldn't really see any effect on the expression of the protein DDX11 that was dependent on CONCR. However, what we saw is that CONCR is able to interact with the protein DDX11. When we pull down the endogenous RNA, it comes associated with this protein DDX11. And we found that this interaction in vitro is able to induce or enhance the ATPase activity of this protein DDX11 in a time and dose-dependent manner, which we don't observe for other RNAs. And in vivo, this translates to loss ability of the protein to bind to the chromatin. So this is a fractionation of the chromatin in a gradient. As you can see here, DDX11 is present in the fractions where of the protein-rich DNA, where the chromatin separated from soluble protein or free DNA. And it colocalized with the London-coding RNA CONCR, but when we knock down the London-coding RNA, the protein shifts to the soluble fractions, indicating that it's not binding the chromatin. And this is also seen when we do chip-seq of this protein in synchronized cells. We release the cells in G1, so we can see when the replication is starting, and we can see that the protein DDX11 is binding at the same locations where the early replication is happening in the DNA. These are replicate data. And when we knock down the RNA CONCR, this binding is specifically, this signal specifically decreases in these regions, but not in the rest of the DNA. So summarizing this work, that what we have found is that in p53 knockout cells, the DNA damage induced expression of a non-encoding RNA that is able to contribute to the enhanced activity of this helicase that is known to help the solving of stalled replication, which is coupled to the establishment of sister chromatid cohesion just after replication of the DNA. So these examples that I show you are non-encoding RNAs that seem to be working on genes or gene locations that are distant from the place where the RNA is transcribed. But we were also very interested on understanding what's the effect of non-encoding RNAs on the location of transcription, which it's actually many RNAs are retained there, non-encoding RNAs, and probably have an effect in the chromatin locally, and also this can has an impact in the expression of transcriptional programs. So to study this, we focus on a cellular process that involves strong chromatin changes. And this is the cellular senescence. Cellular senescence, as you well know, is a stable arrest of the cell cycle that can be induced by different types of stresses, and it's a tumor-suppressor mechanism that cells have to defend from this type of damage. And as I said, it involves strong chromatin reorganization. And involved in this chromatin reorganization, there are protein complexes and chromatin remodelers, as SWISNF complex that is known to be very important in this process. So this complex, SWISNF, it's a multi-unit complex. It has many units, as you can see here, and can have different compositions and conformation depending on what units are in the complex. But some of them are the core units that are present in all of the SWING complex. Like, for instance, one of the core units is SMACRB1, or also called SNF5 of BAF47. Also, this complex is known to be an important tumor suppressor, being mutated in more than 20% of human cancers. So we induce in cellular senescence by using a well-known type of method that is stress by oncogene overexpression. We use overexpression of oncogenic HRAS, this mutant, which leads to proliferation arrest, and also we see like the DNA damage in the cells, beta-galactosidase, and expression of tumor suppressors, which are the typical markers of cellular senescence. So under these conditions, what we did is try to see what RNAs are associated with these chromatin complexes that are involved in the process of senescence. So we use specific antibodies to immunoprecipitate this core subunit, SMACRB1, of the SWISNF complex in proliferating or senescent cells. And we found that a different set of RNAs are associated with this protein under these two conditions. Some of these RNAs are common, but as you can see here in these independent validations, the pattern of RNAs that bind to the protein differ between the conditions, and these differences are not explained by the difference of expression of the RNAs, as we could show by different analyses. And we think that the protein itself also has the ability, this protein SMACRB1, the ability to bind RNA, because when we do this type of experiments under cross-link conditions with UV light, we detect significant association of several of these RNAs. And even though this protein has not, let's say, canonical RNA binding motif, we know that it has a region that is predicted to bind RNA. In fact, that has this motif that has the potential of binding RNA, at least in predictions. And also we found that some patients in cancer have mutations in some of these residues. So also we wanted to know to what regions of the chromatin this complex is binding. So to do that, we use the ChIP-seq method, looking at different marks of the chromatin and also SMACRB1 protein. And what we saw with this is that this SMACRB1 protein that is in the context of the SWINGN complex binds mainly to active chromatin regions, enhancers and promoters. So what we putting together all this data, in summary, we found that there are genes that are bound by SWINGN and others that are binding RNAs that bind this SWINGN from RIP-seq data. And there is a significant subset of these RNAs that indeed are binding, seem to be binding the protein complex in cis, in the same locations where the protein is binding in the chromatin. And we wanted to, as I say, understand this. So we look at one of these RNAs more significantly bound, this London coding RNA that is bound to SMACRB1 in proliferating by not senescent cells. It's an RNA that is enriched in the nucleus of the cell, has a half-life of around 4 hours, and is mostly non-polyarylated. So we named this SWINGN because it's a SWISNF non-coding RNA GAS6 interacting RNA. So this part of this topological associated domain, where we see that this non-coding RNA has the strongest interactions with this protein coding gene called GAS6, as you can see here. And in fact, the RNA, when we knock it down using antisense oligos, is required for the expression of GAS6. So when we inhibit the expression of SWINGN, the expression of the RNA and the protein GAS6 is significantly affected. And this is also dependent on SWISNF. As you can see here, the knockdown of the RNA, of the protein SMACRB1 or also affects the levels of GAS6. So to further test this codependency between SWISNF complex and SWINGN for the regulation of GAS6, we use a natural model, let's say, of the knockout of this protein, which are these type of tumors, rhabdoid tumors, which are a type of tumors that are driven by allelic mutation of the protein SMACRB1. So we found that in cells derived from patients of these tumors, the inhibition of SWINGN doesn't affect expression of GAS6, which is consistent with this idea that the regulation of GAS6 by SWINGN is dependent also of the complex SWISNF. But what happens in tumors where this complex SWISNF is active and is present? For instance, lung squamous carcinomas. Here we see that there is a correlation between the expression of GAS6 and SWINGN, indicating that in this situation we can have a co-regulation. And in fact, this is the case because the downregulation of SWINGN affects GAS6. GAS6, as you probably know, it's a protein that is secreted to the medium and binds to tyrosine kinase receptors, activating signals for proliferation and survival of the cells. So if SWINGN is upstream of this oncogene, this probably has effects in these pathways. And of course, what we find is that the downregulation of SWINGN results in less proliferation of the cells and also less capacity to form tumors in mice. So far, what I show you is that this London-coding RNA, SWINGN, interacts with the protein SWISNF to regulate GAS6. However, we were suspecting that maybe there are other targets of this RNA. And the reason for that is that the expression of GAS6 is not sufficient to rescue the phenotype that we observe when we knock down SWINGN. So we propose that maybe other genes could be also co-regulated by SWINGN and the complex SWISNF. And supporting this idea is that we found that the vast majority of genes that are regulated by SWINGN also are bound by SMACRB1, so they are targets of SMACRB1, and also they have the typical motifs in their promoters for SMACRB1 regulation. So to see if there is really a dependency in the regulation of these other genes, additional genes, we knock down SWINGN and analyze the binding of SWISNF or SMACRB1 to the chromatin. We didn't see a general effect, but we found that in a subset of genes, the binding of SMACRB1 is affected. And in fact, the majority of these genes, they have less binding of SMACRB1. And this correlates also with a decrease of the lysine-27 acetylation. So there is an effect in the chromatin state of these genes that are co-regulated by SWINGN and SMACRB1. This is a set of these genes, this signature of genes that are co-regulated by SWINGN, SMACRB1, and they also have this change in acetylation of lysine-27, as I said. And for instance, one of them, of course, is GAS6, which we already knew, but there are other interesting genes like this PDGFR-beta, which is one of the known most important targets of in teratoid tumors, in these atypic rhabdoid tumors. So confirming that these genes are regulated by SWINGN, we can see that if we, instead of knocking it down, we can upregulate it by using the CRISPR-A technique. This also results in the upregulation of these gene targets. And we know that SWINGN is physically binding some of these genes, because when we do CHIRP, which is a technique that use RNA probes or DNA probes to pull down the RNA bound to the chromatin, we find that SWINGN, here in red, it's associated not only to its own promoter, but also to promoter of the regulated genes that like GAS6 and also PDGFR-B. So this regulation of several genes is necessary for this phenotype that we observe in the cells. If it's an oncogenic signature, because we found that the combined downregulation of more than one target of SWINGN increases the phenotype of the cells when we inhibit them. And when we look at their expression, the expression of this signature in patient samples from squamous cancer, these the patients of cancer, they clusterize based on this signature, and finding that while the normal non-tumoral samples have lower levels of the signature, the tumor samples have much higher levels, confirming that is indeed SWINGN is defining that tumor an oncogenic signature. So what we have found is that this long-encoding RNA SWINGN interacts with this complex SWISNF to regulate GAS6, which is a gene that is in the same topological domain, but also can regulate other genes that are distant, that some of them are even in different chromosomes, and it's able to contribute to their activation. So when this occurs in proliferative cells, in cells with lower proliferation, like in senescence cells, this interaction is not occurring, and this induction of expression that is favored by SWINGN, this is not occurring. So as a general conclusion of all that I show you, like very general ideas is that we know there's a lot of things that we still don't know, but what is very clear from the work of many people, including us, is that transcriptional alterations of LINC-RNAs are inherent to cancer cells, and these alterations contribute to the phenotype of these cells. So they have the potential of being interesting therapeutic targets. Some of these LINC-RNAs, they not only control gene expression, but also genomic instability, like CONCR, that regulates chromatin cohesion. And also, we found other examples, like SWINGN and possibly others, that contribute to transcriptional activation of promoters from enhancer loci. So just want to thank the group, my lab, that did the work, especially Francesco, that he did the work of CONCR, and Elena, that did the work on SWINGN, and also collaborators and the source of funding. And thank you.